Hello students, welcome back to the channel. In this video, we are going to study generation of the nerve impulse. But before generation of the nerve impulse, try to understand how many types of channel proteins are there. Now see, electrical signal which is produced by the neurons and the muscle fiber depend upon different types of ion channels. First is leaky channel which will be open and getting closed randomly. So randomly it will open and it will get closed. Another is voltage gated channel. Voltage gated channel will open with respect to the voltage present inside. For example, inside the cytoplasm, if it is minus 70, the gate is closed. It will be open only when the voltage become minus 50 millivolt. Third type of channel is ligand gated channel. Ligand means chemical. Any chemical, for example, like acetylcholine or GABA, any chemical neurotransmitter. Here you can see if any chemical bind, because of the binding of the chemical, the gate is open. We say that is ligand gated channel. Now see, best way to learn the neuron is salty banana. This is the learning tips. Salty banana. The salt means you know very well with the sodium chloride. So there are three sodium chloride outside, means just giving an idea. And banana is rich in potassium, you know very well. So potassium is inside. It means outside the neuron, sodium concentration is high, and inside neuron, potassium concentration is high. Clear? Not only these two ions, some other things are also there. For example, if you see, negative charge proteins will be present, and chlorine will be more outside, calcium will be more outside, protein will be very less outside. So this is the way how unequal distribution of the charges will be there. Now see, for the generation of conduct and conduction of nerve impulse, try to understand three things. First is resting potential. Then we'll be talking about action potential, in which depolarization of neuron and repolarization of neuron. And third, refractory period. The resting potential is the one in which the membrane is having a potential, and you can say it is polarized and it is ready to receive the stimulus to start the nerve impulse. Now, how do they achieve the resting potential whose value is minus 70 millivolt? They are able to achieve the resting membrane potential because of unequal distribution of ions. Outside already we have seen that sodium concentration is high. Okay, the sodium concentration is high outside. You can see in this diagram, sodium ions are high outside. And inside you can see here, this is the symbol used for potassium ions. So potassium ions are high in the cytosol. Clear? The extracellular fluid is rich in sodium ions and cytosol is rich in potassium ions. Clear? Now, not only sodium ions are present high, you will find here in the cytosol, negative charge phosphate group is also present, and few proteins, big size proteins, they are not able to leave the cell. So not only unequal distribution of ions, inability of the anions to leave the cell, this is also responsible to create a potential difference. Third reason is the sodium potassium exchange pump, which is expelling 3 sodium out and bringing 2 potassium inside. So it is trying to create a gradient. This is a not only concentration gradient, even charge gradient also. Sodium is high outside, clear? Chlorine is high outside. Potassium is high in the cytosol and protein are also high in the cytosol. So if you see, not only charge gradient is created, it is a concentration gradient is also created. We say electrochemical gradient. Now, on the arrival of a stimulus, what will happen? Action potential will be created. Action potential is also known as nerve impulse. So once the depolarized graded potential is created, that is nearly minus 50 millivolt, which is a threshold, what will happen now? Voltage gated sodium channel will open. Voltage gated sodium channel will open. Please understand carefully. For resting potential, we were working with the leaky channels. Let me show you once again. For resting potential, we were working with the leaky channels. And leaky channels, if you see, more were present for the potassium ions and less were present for the sodium ions. Okay, this was for sodium. So these are leaky channels. Okay, these are leaky channels. Because of this leaky channels, resting membrane potential was created. Now we are discussing about the voltage gated sodium channel, which will open when the stimulus reach to the threshold potential, that is minus 55 millivolt. And because of the high sodium out, less is inside, an electrochemical gradient is there, because inside, earlier it is minus, okay, earlier is minus and outside is plus. So not only concentration gradient, even charge gradient favors the movement of sodium inside, and as a result, inside becomes positive and outside is negative. We say now the membrane is depolarized, or you can say action potential. Clear? Now, after action potential is done, what will happen? This is a sodium voltage gated channel which will get closed, and potassium voltage gated channel will open, because of which potassium will go out. Is it clear? Potassium will go out, and once the potassium goes out, what will happen? Slowly, this area will become now positive. Is it clear? Because of the opening, it becomes positive, and we say it is repolarized. Repolarized. Now that what is refractory period? The period of time after an action potential begin, during which excitable cell cannot generate another action potential. We say that is refractory period. Because in this case, what has happened during resting potential, let me draw and explain you. Here what has happened, potassium concentration is high outside, sodium is low. Inside, sodium is high and potassium is low. So if you see, polarity came back, but ion distribution is not correct. Ion distribution is disturbed. For resting potential, what was the condition if you remember? For resting potential, sodium was high outside and potassium was high inside. So it goes for short refractory period in which sodium potassium exchange pump will work and expel 3 sodium out and bring 2 potassium inside. So I hope so this video might, up till here, this might be helpful for you. Now see the summary. Try to understand. This is the stimulus site, which was a membrane, was at the resting membrane potential, means minus 70 millivolt. Now when the stimulus is coming, it is going to the threshold potential. Threshold potential is minus 55, and because of which sodium get open and rapidly depolarization is done, and the value goes to the plus 30. Now what will happen? Polarity will be reversed. Repolarization. Is it clear? Repolarization. So here what will happen? Here after this phase, sodium gate will close and potassium gate will open. But potassium voltage gated channel take a time to close. Sodium immediately close, but potassium will take time. So it goes for hyperpolarization. You can see here hyperpolarization phase. This is a simple summary, same diagram, understand it. This is threshold potential. Clear? Sorry, just 1 minute. It's not a threshold. This was resting potential. This is your resting potential minus 70. Once it goes to the threshold, then what will happen? Sodium gate will open. Okay, sodium voltage gated channel will open, and in this phase, immediately it is going to go for depolarization, becoming plus 30. Clear? After it becomes plus 30, it has gone for depolarization, sodium channel will close, and at the same time, potassium channel will open. Now, potassium channel will take time to close, so that is why it goes for hyperpolarization. Clear? So, if you see during resting membrane potential, Sodium potassium exchange pump was working. Passive diffusion was going on through leaky channel. Voltage, sodium voltage gated channel was not working. Potassium voltage gated channel was also not working and value was minus 70. Means inside was negative. During depolarization, you can say action potential. Pump is not working. Passive diffusion is still going on. Now voltage sodium will open but potassium will not open and the value become plus 30, means inside is positive. During repolarization, pump will work. Passive diffusion will continue go on. Sodium voltage gated channel is not working. Only potassium voltage gated channel is working, which is responsible for hyperpolarization also. It makes the value again to minus 70, but because of hyperpolarization, it is minus 85. So I hope so this video has helped you to understand what is resting membrane potential, how it is achieved, what is action potential. Action potential has got two phases. First is depolarization, then second is repolarization, and it wait for the short period that is known as refractory period. In the next video, we are going to study how the action potential is propagated. Once it is created in the one side of neuron, how it is able to reach to the another end of neuron by the local current that we'll be discussing in the next video. I hope so this video might be helpful for you. Thanks for watching.